Hello everyone, myself Dr. Vivek Kumar, Professor, Himalayan School of Biosciences, Swami Rama Himalayan University, Joli Grant, Dehradun. I am again with you in another subtopic of endophytes, that is methods for studying endophytic bacterial diversity. So endophytic bacterial communities, they are conventionally studied using culture-based methods. Culture-based method means you have to culture the endophytes which you have isolated from any sample. So however to investigate the endophytic bacterial communities using these methods? So bacteria must be cultivable under laboratory conditions. Cultivable means whatever the microorganisms you have isolated, it should be alive and should be able to show its growth on the petri plate. So cultivation process relies on the isolation of endophytic bacteria from the plant tissues. Here the plant tissue can be any part, as I have told you before, it could be root, stem, leaf, seed, flower, twig, anything. For isolation, the isolation procedure, it should be sensitive enough to recover most of the cultivable endophytic bacteria. Sensitive enough means our technique should not kill the endophytes. Our technique should be able to isolate maximum number of microorganisms, maximum number of endophytes with a maximum number of diversity, but should be strong enough to eliminate epiphytes. Epiphytes means those microorganisms which are present on the surface. If we are not able to remove those microorganisms from the surface, then we will have a false impression that we have isolated the endophytes, but in fact we have not, and other contaminating bacteria from the plant tissues being processed. So normally the isolation protocol requires surface sterilization of plant tissues followed by their maceration, crushing, etc. Then whatever the sample we have got, then serial dilution of that sample, and then plating on the bacterial growth medium. In this way, generally we have seen that if we use different cultivating medium, normally people use Newton agar cultivating medium, then that will give us more results in isolating more different types of endophytes. This picture shows that here there is a plant is there, and for example you have taken root, or you have taken a stem and twig, or you have taken leaves. Then you are using sodium hypochlorite, you have sterilized it. Then after that you have sterilized using ethanol. Then you have given several washes using sterile distilled water. 6 to 7 times of washings are required to remove any traces of sodium hypochlorite and ethanol. Then after that you have cut down the pieces of their surface samples into small pieces, and you have put on the media, and then you have incubated your petri plates. Then after some time you will observe the growth of microorganisms around your sterilized samples. Okay, so this is the very common procedure. Then after that, once you have seen the microorganisms, you have to isolate them, you have to purify them, you have to identify them, and you can keep them after testing them if they have some potential plant growth promoting attributes. So in this process, the sterilizing agents like sodium hypochlorite, ethanol, hydrogen peroxide, they are commonly used for the surface sterilization. And these chemicals are used in a series. For example, sodium hypochlorite, then we can say go for the ethanol, etc., to improve the effectiveness of the sterilization process. And then, as I told you before, after the sterilized tissue is washed several times with the sterile distilled water to remove any residues of the traces of the sterilizing agents. So sterilization is confirmed by plating small amount of distilled water from the last wash. As I have told you, 6 to 7 washing. For example, if you are going to take 6th wash or 7th wash, then the last wash you have to take and then wash that sample of that you have to spread on a petri plate on a suitable medium. If that spreading after incubating for 24 hours, 48 hours, say maximum 72 hours, if you do not observe any growth, it means the process of sterilization was perfect and complete. Now whatever the microorganisms you will get from your samples, they will be certainly endophytes. Again this picture shows, this will make you better understand. You have taken sample, leaf sample, you have sterilized them on this is the control plate, you have not inoculated anything, and here after sterilization you have put your leaf tissues on the petri plates, and then after incubation of some days, some time, you can see the growth of microorganisms from the samples. The surface sterilization process, it producing high number of endophytic bacterial growth on agar media indicates minimum damage to the endophytic population or community by the sterilization process. So this is desired, as any damage to the endophytic community by sterilizing agent can compromise the authenticity of the bacterial diversity analysis. If our process is going to kill the number of endophytes, then we are not going to get the desired results. So these cultival bacteria or fungi that are identified using morphological, physiological, biochemical, molecular approaches, where the molecular approaches are considered best as compared to the other approaches. Again, this picture shows that you have taken the plant samples, and when you have sterilized the sample and put sample on the Petri plates, then you can see the growth of microorganisms on the Petri plate. So these microorganisms, after proper sterilization, if we observe on the Petri plate, it means these are the real endophytes. And then a number of molecular markers have been identified that permit the identification of spatial microbial taxa and their phylogenetic classification. Among these molecular markers, 16S ribosomal RNA is the most commonly employed to identify bacteria and determine their phylogenetic relatedness. We have used 16S ribosomal RNA because the sequence in 16S ribosomal they are conserved. Conserved means since the time of evolution there are little or no change. So we believe that these are the conserved sequence. So every organisms, they do have their own conserved sequence. So we can compare them each other. So we can, analyzing the 16S ribosomal sequences, we can compare the different organisms to check the evolution or the relatedness among the microorganisms. So the total population estimates of endophytic bacteria in plants may vary. So these estimates can depend on the type of the growth media used. Sometimes you are using media A. The media A is, if you have used it, then it is nutritionally poor, then obviously it is not going to support the good growth of all the endophytes. So we have to use different types of, 3-4 types of cultival media. So one media will support some bacteria, one will support another bacteria. So this you will give a holistic amount of bacteria, good number of bacteria you will isolate from the tissues. For example, a successful surface sterilization can result in the penetration of these sterilizing chemicals in the interior of the tissues. Once they make the entry inside the interior of the tissue, then obviously once they have killed the surface bacteria, they are also able to kill the inside bacteria. So killing the endophytic colonizers and compromising the correct bacterial estimates, this will be a problem. Now there is a role of growth medium. So similarly, selection of growth medium also affects the numbers and diversity of endophytes that can be isolated from a specific plant tissue. So we can understand that as no one medium can meet the nutritional and growth requirement of all the bacteria. So to cultivate a large number of bacteria, we have to use different types of growth media. So growth media are not always a reason for the to culture bacteria, because some of the bacteria, they are viable but non-culturable state. So when they are viable, not culturable means we are not able to cultivate them. We are not able to give them those nutrients which are required for their growth, not able to show their growth on the Petri plate. So when the endophytic bacteria have been successfully isolated, so maintaining of the growth media can sometimes prove to be difficult. You isolated, but they are not, after some time they are stopped growing. It means initially the nutrients they utilize, but later on for their multiplication, the desired nutrient is not there. So it is recommended that endophytic bacteria isolated using more than one type of growth media and supplementing this media with the plant extract. Plant extract means the same plant from where you have isolated your endophyte. Why? Because that plant extract will give those nutrients which are required for the growth of the endophytes, because the endophytes, they are acclimatized to a particular environment, the particular nutrients which are present inside the host plant. If you provide them in the Petri plate, obviously the bacteria or fungi will show their growth. However, cultivation dependent methods can strongly underestimate the number of bacteria present in the plant tissues. So culture independent methods to study the endophytic diversity, so they rely on total bacterial genomic DNA extraction procedure. So plant tissue is first processed to remove surface microbes, So usually achieved by aseptic peeling techniques to remove the surface layers, vigorously shaking the plant tissues with acid wash glass beads in saline solutions to dislodge the surface bacteria, followed by wash with sterile distilled waters, and then process plant tissues then homogenized to extract the bacterial genomic DNA. The genomic DNA then can be analyzed using a range of molecular fingerprinting techniques. The most commonly the genomic DNA I told you before is the 16S ribosomal RNA gene to analyze any bacterial diversity. Now there are different novel techniques. The variety of amplified gene fragments representing the entire endophytic population of a plant are then analyzed using community DNA fingerprinting techniques like amplified ribosomal DNA restriction analysis ARDRA, denaturing gradient gel electrophoresis DGGE, temperature gradient gel electrophoresis TGGE, and terminal restriction fragment length polymorphism TRFLP. These are the different techniques, novel techniques to analyze the sequence of the endophytic bacteria. Alternatively, the highly variable regions between 16S and 23S DNA, ribosomal DNA, they can be analyzed automated sequencing using ribosomal interspecies spacer analysis (ARISA) for the community fingerprinting, and to be detected by these fingerprinting techniques, an endophytic population must represent about 1% of the total community. Moreover, the chances of detecting novel bacteria using these methods are low. Why? Because the databases of fingerprinting methods are largely incomplete. And then there is a metagenomic approach also. So the DNA fingerprinting techniques have largely been superseded by more advanced molecular techniques like metagenomics to study the microdiversity. The metagenomics involves DNA extraction from the entire bacterial population for analysis of gene using next generation sequencing. The sequencing could be done for the entire DNA, which is then assembled and annotated to understand the functional sequence, functional part of genome, or it could be done for one particular gene or polyphyletic marker like 16S ribosomal RNA. So the metagenomic approach allows full depth of endophytic diversity analysis in comparison to traditional fingerprinting. So using metagenomic approach, hidden community of rice endosphere was uncovered in one research, and in this research they have deciphered many traits which have been shared by endophytic inhabitants that might be crucial in their endophytic competence and success. However, the DNA based community analysis cannot selectively analyze the viable or metabolically active bacterial cells. For this, RNA based approaches are generally utilized. So these can specifically determine the metabolically active population, as the amount of RNA can be correlated with the growth activity of the endophytic bacteria. So because the metabolically active population, they will show the more amount of RNA. In one study, rhizosphere bacterial communities of 3 related legumes plants were compared. So it was observed that metabolic profiles of 3 bacterial communities were more dissimilar. They showed 45 to 50% similarity as compared to DNA based profiling 70 to 90% similarity. Similarly, comparative messenger RNA based and DNA based analysis of root associated communities in rice plants revealed that only a fraction of nitrogen fixing bacteria actively performed the activity. So endophytic bacterial population can also be studied directly in their natural settings. The techniques like fish fluorescence in situ hybridization, they have allowed studying the endophytic bacteria in natural habitat. So moreover, by combining fish with another DNA fingerprint technique, so the dominant population of endophytic community of plant can be easily identified. Such a polyphasic approach that combines different approaches is recommended when analyzing the endophytic bacterial or endophytic microbial populations. This picture shows the fish technique. You have a mixed population, then you fix the population microbiome, there is a hybridization, then you have a fluorescent dye with a probe DNA, then the probe of DNA having fluorescent dye will go and attach to the, we can say, the similar sequencing, and then it will hybridize the cell. Using this fish technique, you can identify that particular sequence. Again, this thing, there is a DNA probe. You have, a DNA probe is labeled with the fluorescent dye, and then you have denatured and hybridize. The hybridize with the go with a similar genomic sequencing. In this way, you can identify the particular sequence. Finally, if we discuss the key points of today's lecture, the isolation protocol requires surface sterilization of plant tissues followed by their maceration. Maceration allow the release or separation of the, we can say, the endophytes from the tissue part, then serial dilution, and then plating the bacterial growth medium. So as we discussed, we have to use different types of growth medium. The genomic DNA then can be analyzed using a range of molecular fingerprinting techniques. Genomic DNA of whom? Endophytes. The most commonly the genomic DNA is used to employ marker gene. We are going to use 16S ribosomal sequencing. Using metagenomic approach, the hidden community of endorhizopsore may be uncovered. So, thank you so much. We will meet in another lecture on endophytes.